started. And maybe in a minute you can, well, people are still going to come in, so in a few minutes we can shut the door or something. So, I'm Jonathan Eisen. I'm at UC Davis and also have an adjunct position at JGI. And my grand vision for this session was to tell everybody to leave and to go to all the other sessions, because I actually think it's really important for the computational people to be integrated with the scientific questions that are being asked. But I'm not going to do that. You're welcome to get up and leave if you decide that you want to do that. But I do think there are some useful things to talk about among the computational people specifically. So I was convinced not to do my radical plan of sending everyone away. But I do think that one thing that's somewhat missing at this sort of type of meeting is a chance to get to know what a lot of other people are working on. And so I would like to take the first few minutes for people to just say what they're interested in terms of just a couple sentences, who they are and what they're interested in. And at least that way, if there are people interested in the same things, you'll find that out and you can talk about it later, or from the sign-up list, maybe they'll post that. So I'm sort of generally interested in evolution of microbes, and in particular how new functions originate. But mostly what I've been doing for metagenomics is just the first step, which is trying to use metagenomic data to look at, reassess who is out there from microbial samples. So looking in the metagenomic data for phylogenetic marker genes and seeing if you get the same answer as with ribosomal RNA PCR studies. But I've also been doing a bunch of other computational metagenomic studies. So if you don't want to say anything, don't say anything. But if we can just go around and just introduce yourself and say briefly what you're interested in.
So my name is Konrad Förstner from the PABO group at EMBL in Heidelberg, Germany. And I'm doing biomining for biotechnologically important
I'm Mihai Pop from University of Maryland, College Park, and we work primarily on genome assembly in the context of metagenomics, also use sequence technology. We can use this to do metagenomic research.
My name is Trin Wang from Stanford, and I'm interested in looking for viral sequence from the. We use 4x4 to sequence sample from patients. Blood, all kinds of samples. We try to look for new virus use computational tools.
I'm Kevin Penn, and I'm a grad student at Scripps, and I'm mainly looking at the genomes of environmentally isolated actinobacteria. So we have 2 genome sequences for these, and been studying the ecology and evolution of these 2 bacteria based on their comparative genomics of these 2.
I'm Beltrán Rodriguez. I'm from STSU. I do metagenomics involving prey-predator relationships in phage and microbes to try to figure out who and what are they killing each other.
I'm Rotem Sorek. I'm AGI. Interested in antimicrobial proteins, horizontal gene transfer, and non-toxic RNA.
I'm Saul Kravitz with the Venter Institute, just generally interested in metagenomics tools.
I'm Sheila Podell with the Scripps Institution of Oceanography. I've been doing a lot of genome analysis, annotation, and mining, and I've just recently switched to be looking at metagenomes. I'm particularly interested in horizontal gene transfer.
Okay, I'm John Hooper from JGI. I'm interested in binning and clustering mostly.
Thomas Iketsfontein from Technical University in Denmark, Søren Brunings group. We are mainly interested in machine learning approach combined with metagenomics data and horizontal gene transfer detections.
I'm Dawn Field from the Oxford Center for Ecology and Hydrology. My background's in molecular evolution, so I'm interested in large-scale comparative studies, for example, especially orphans, trying to understand why they have these genes, looking for trends, and so we've gotten very interested in standardization, how you get more data attached to the actual genomes or metagenomes.
I'm Victor Marcos from Lawrence Berkeley Lab, and I'm interested in the large-scale data management and analysis.
Nikos Kirpidis, JGI, interested in genome and metagenome analysis, tool development and factual analysis.
My name is Ken Klopo from NIST Japan, and I'm very interested in genome analysis and metagenome analysis of human gut microbiome.
I'm Anthony Fodor from the Bioinformatics Resource Center at the University of North Carolina at Charlotte, and I'm interested in community composition of freshwater and wastewater and metagenomic analysis for discovery of this.
I'm Robert Olson from Argonne National Lab. I'm working on the SEED project there, genome database work. We're also starting doing metagenome analysis server work.
Tim Mcpiller, and I'm involved in development of sequencing and computational technologies for all the health of this field.
Claudio Farfas from Collective Tools, software and system architectures, and system integration tools, so the computational part of the decision hunt.
Yeah, my name is Daniel Dalevi, and I work with IMG, and I'm interested in metagenomics tools in general.
My name is Marcel Huntemann. My background is also binning and clustering, and I just started to work for Jonathan at the JGI. Try to help him answering some of these questions, and as you most probably know, he has a lot of questions.
I'm Sarah Light. I work at Lawrence Livermore National Laboratory, and I'm interested in predicting phenotype from genotype, generally speaking.
I'm Bill Feiereisen. I'm from Los Alamos National Labs, and I'm interested in large-scale data analysis and metagenomics tools, particularly oriented around pathogens and possibly around biofuels.
I'm David Sorgel. I'm at Stephen Brenner's group at Berkeley. I'm interested in sequence composition-based binning, as are a lot of other people taking the credit.
I'm Jing Chan from In Silico Discovery, and we build data integration software and create software for helping users to create ontologies and query the ontologies they create.
I'm Judith Cohn from Los Alamos National Lab, and I started off in genomics working for JGI at Los Alamos, and on the Human Genome Project for the last 4 years I've been working in protein function inference, and I'm just getting started with metagenomics now.
Okay, my name is William Xiang from Simon Fraser University. Just finished my PhD in studying genomic islands and horizontal gene transfer, and will be going to Claire Furcher's group in October to study metagenomics, to start metagenomics studies.
My name is Saurabh Chatterjee. I'm a postdoc with Jonathan Eisen at UC Davis. I'm interested in metagenomic analysis in general, but right now I'm focusing on the binning problem.
My name is Anne Bergeron from Montreal, Canada. I'm interested in the mathematical aspects of experimental design and assembly.
My name is Krishna Subramanian. I work at the Joint Center for Structural Genomics, study protein evolution by looking at protein structures and classifying them.
I'm Rachel Lee. I'm local here at Coventry to work with camera project. I've been working to identify new genes and the protein, and also a protein family classification and identify new protein families from huge metagenomic data sets through faster clustering algorithm. Mostly interested in any large scale sequence analysis.
So if you can't tell already, there's a small group of people, but pretty diverse questions and or interests here, which is standard for metagenomics, I mean for genome analysis in general, but metagenomics is probably even more diverse in terms of the questions people are asking, because it's opened up all these questions about ecology and distribution patterns of organisms that really weren't possible with other genome sequencing methods. So I sort of have an outline. I think it is somewhat useful for people in the field to take a step back maybe and think about the general scope of the field, or maybe what the challenges are in the field. And I'm sure 90% of the people here are more appreciate some of this, so just throw things at me. But sort of, these are just some of the challenges. But I mean, I think that these help guide why it's hard to develop methods to do metagenomic analysis, and why we can't just take methods that have been developed for analyzing single organism genomes and immediately run them on metagenomic data, even though that's what many people do, because we don't really have a choice in some cases, but we haven't yet figured out which of those methods work. So obviously the fragmented data, in this context I mean short sequence reads that aren't part of whole large contigs necessarily. So a lot of the methods designed for analyzing isolate genomes are based on the idea that at least you have a large collection of data from the same organism. Even better if you have contigs or closed genomes from the same organism. Similar issues come up with other aspects of the fragmented data, but really it's the short sequence reads of, from samples of the organisms. And of course the mixed organisms in the community raise a lot of issues that I'm not sure have been completely dealt with, although some people claim that some of them are solved. I'm not sure that that's really true. So even gene finding, which is still a challenge for isolate genomes in some cases, when you try and do gene finding from a complex community of mixed organisms, with each with their different GC contents and codon usages and patterns of variation within the genome, how we actually do a good job with finding genes is going to be complicated. And the same is true for a lot of other methods. When you design a method that works for a single organism's genome, it just may not work that well when you're running it on a mixed data set of a community of organisms. You might be able to optimize those methods if you had any idea what the community structure was like for your sample. But in many cases, we don't. In some cases, there's been prior research with ribosome RNA, PCR, and ERISA, and T-RIFLET, and sequencing, and in situ hybridization, and the other things like with stuff Jeff Furman was talking about with some of the human samples. But in a lot of cases, people are doing metagenomic analyses as the first step in characterizing a sample. And so there may be nothing known about the community. And so the method that you use, a lot of them, it may not even be the settings, it may be the actual total approach that you use depends on the balance between the total number of organisms and their phylogenetic distance between them and what types of organisms are there. So when we don't know what the community is like and what those genome structures are like, it can be very challenging to optimize the methods. And of course, if you want to do comparative analyses, so comparing multiple samples to each other, if you have to fine-tune the methods that you use to the community structure, and then all the communities have different community structures, how you then compare those different results to each other can be really challenging. And I think, I'm not sure who gave the example, maybe it was Janet today who said that she took the tiger data set from the human gut and then couldn't really analyze that data set in her context and waited for IMG to reprocess the data and then use the IMG reprocessing of the data. And I know that one of the things Nikos tries to do, or the people, Nikos and Victor, with IMG is to standardize what gets into that data set. But of course, if we decide that in the end a low diversity ecosystem has to be treated differently than a high diversity ecosystem in terms of the methods that are used, how do we normalize between them and standardize? It's going to be a challenge. And the same is true for genome structure. So doing this for, everybody keeps saying we're not doing those protists with metagenomic, those nasty eukaryotic things with large genomes. But it's going to happen. There's going to be a lot more data from eukaryotic microbes from a lot of samples. And we're already seeing this with some of the plasmid surveys that have been done and the viral surveys. We can't sort of assume some type of standard genome structure within the samples. And then, of course, something that also has not been really dealt with very well, or will continue to raise its head, is that the methods will have, the samples that you're analyzing will have been gathered with very different methods, the data. So that includes how the DNA is actually collected. We've heard some examples of that. It also includes how the DNA is sequenced. And then if you want to compare 20 samples that are all been gathered with different collection methods and different sequencing methods and try and normalize across those samples and then correlate this to some result to the metadata that's associated with those samples, it's going to be very challenging. I mean, Dawn sort of pointed this out with saying that we at least need to know what the methods are used, but that doesn't, of course, solve the problem. That just at least gives you a bit of information that you can actually try and ask the question how to solve the problem. But I mean, even how we join 454 sequenced samples to Sanger sequence samples and compare them to each other, I don't think we really have a good handle on, and how we design computational methods. So I think that for the big picture, for the future of any method in computational metagenomics, you need to sort of think about what the samples are going to be like that are coming, what the variation among those samples is going to be, and then try and imagine how to deal with that. And I mean, there are many approaches to doing this. One approach, which I think is really powerful and has not been used extensively, but you see individual examples of this, is to do more simulations in the context of doing your testing your metagenomic method. And Nikos had a paper in Nature Methods or that just came out on sort of using simulations to test a few different aspects of metagenomic methods. There have been examples within the context of genomic studies and ribosomal RNA studies. Jed even gave an example of this today, where people have done some simulations in the context of their analysis. I think we desperately need more of that in the context of answering these questions. How do you combine 454 data and Sanger data, et cetera? And so what I basically did is, I'm sure there are other challenges. I don't know if people, am I leaving out some of the specific things about metagenomic data here that people think are related to sort of the grand challenges of analyzing it? I don't know if I've captured everything. Please jump in if I...
Updating of databases in terms of concurrency and changes in frequency with which databases are updated, and getting data from different sources. In terms of inconsistency, there's not only a diversity of where they came from, but how often they were updated and...
Yeah, I didn't say that because I think that that's a general problem, not a metagenomics, right? So that's been a problem in all the genome sequencing projects as well. So I tried to focus on specific. There are a lot of other issues with metagenomic data that are in parallel to what has happened in genome sequencing or other sequence-related fields. So that is definitely a huge problem, and this is part of Dawn's initiative, is to build database structures and encourage the community to get involved in adding that information, such that you don't need that a lot of information is there at the beginning, right? But in addition, if something gets re-annotated, right? I mean, this is a standard problem in genome sequence analysis, is keeping up with all of that. I was trying here to focus more on what is different about metagenomics that makes it even harder than dealing with some of those problems. Yeah.
One point you might add is just the pure size makes it.
Yeah, I forgot that. Yes, absolutely. So.
Different normal genome analysis.
Although there are, there are examples of, when they sequence the amoeba genome, the total massive sequence data will be large. So yes.
Yeah. I'm going to disagree a little bit back to the point, because one difference is not just the information that we have an updated database. It's the fact that your data is only meaningful as it's compared. And so the historical data is only meaningful as it continues to be compared to new data. And it's complex. That wasn't necessarily true in the genomics work, because you kind of did an assembly. Right? You could actually stop de novo, do a new assembly, which is what we did, right? In this case, it really is a very evolving relationship you have with your data versus other data. And that's a huge complexity and this computational science problem that we didn't really have in the genome.
Definitely.
And also, I think we need to have some kind of minimum requirement for the amount of the sequence to be acquired for a given specific sample. What I mean is, for example, with the organismal genome project, the criteria is kind of clear, so you know when you're finished. But with metagenomic data set, oftentimes the problem is it gets open-ended. So You don't know.
But I think that you're going to just have to suck it up and accept that that's the fact.
Well, what I mean is, for example, if you know the complexity, or if you can guess the complexity out of the genome data based on, for example, 16S RDNA sequence data, then you can estimate the coverage. For example, you have 10x or 20x.
Yeah, no, I understand that there would be a goal for that, but I mean, as David Relman talked about, a lot of people are interested in the rare organisms in the sample, and you're certainly, I mean, if you set a criteria for those rare organisms, it would never be met, right? I mean, it just so you have to, I mean, and each community, I mean, this is the problem. This goes to the variable community structures, is that each community is going to, in fact, be different with that approach. So for the symbiont communities that I've worked on, and that like the gutless worm symbiont community, or Jill Banfield's acid mine drainage, where there's relatively low diversity, I think you might be able to invent parallels to the genome sequencing criteria. But for soil or the ocean, where an organism that's present in 1 over 10 to the 6th cell, really rare organism that might still be important, I think it's harder to come up with.
I understand your point, but what I'm trying to say is, for a given environment, you could sequence, for example, millions, or you could sequence just 100 or several hundreds of sequences, and still you say it's a metagenomic project.
Yeah, and what I'm predicting is that's just the way it's going to be. People will be publishing papers where they did 10 reads. If they found something interesting, they're going to call it a metagenomic study.
It depends on your question. If your question requires 100 reads, why do more?
Yeah.
Well, that's true, yeah.
No, but I mean, this issue has been, TG raised this in his talk. I think that, for their metagenomics initiative, to get the government agencies interested in funding something, it can't be open-ended, right? They need to set, as a community, what is the goal here. But for any individual sample, if it's a complex community of microbes, setting that goal separate from a scientific question is very hard. For isolate genomes, it's very easy. The goal, you can just say the goal is to finish the genome sequence of this isolate. And then you may not want to do that for amoeba or some weird protist genomes, but you can still say that could be a goal. And it's just much easier to remove that from the scientific question evaluation. So I think it's and so what I basically did is I think I
think one of the big problems for metagenomics is the validation. When we develop new software, new method, and we make predictions of them, how to validate.
Yeah, I will get to that. So I absolutely agree. I don't consider that. I mean, again, what I was trying to talk about is the data sets themselves, what's different about them. But absolutely, the fundamental thing we all need to do is to deal with evaluation of the methods, which is also true in analyzing and in doing any computation. And so one thing that's common, right, is to take a method that was designed for an isolate genome or something else and try and design a method to adapt the method to deal with that. There's assembly methods, there are annotation methods, there are comparative and evolutionary methods, we've heard examples today of functional genomic methods. Many of them don't work ideally for metagenomic data sets. And the question really is how, for computational people, is can you adapt those methods, or do you need to start from scratch and invent new methods? And I don't think, I'm sure people have ideas. I don't really work on a lot of these things about the relative balance. I think that there are going to be some cases where you can just adapt an old method. For functionally annotating individual sequence reads, I think you can just adapt the methods that were used for either annotating ESTs or annotating genome data in a large part, and removing all the methods that would use context as part of annotation. But for predicting pathways in communities of organisms where you have 5% of the genome sampled of one organism and 1% of the genome of another organism sampled, how do you actually make any prediction of pathways in that case is very different than what people were doing for genome sequencing projects. I mean, the thing I'm most interested in myself, although I know that it's not the case for everyone here, is what are the things we can do with metagenomics that we couldn't do with other approaches or other methods? And here, the computational methods, some of them are pretty straightforward and some of them are not. And so, for example, the standard question that everybody's asking about environmental microbiology, who is out there, we can start to reevaluate with metagenomic data. And so we've seen we've seen examples at this meeting and at lots of other meetings where people have found something in metagenomic data that they did not, just in terms of the types of organisms that are there, that was missed with previous methods. So some lineage that ribosomal RNA PCR did not amplify, or some lineage, or, well, virus. I mean, right? So forget about any of those. Just basically anything about viral diversity has been pretty much revolutionized by metagenomic sequencing because PCR approaches generally didn't work for serving broad viral diversity. And so in terms of this who is out there, there are two things that actually metagenomic data can do, and I think there are lots of interesting computational challenges there. One is just asking what are the phylogenetic types that are present in a community. I think we can do this differently because we can do this with things other than ribosomal RNA. That's why you can find viral diversity, and that's why I think we will find new cellular organism findings because we can look at other genes. But there's also microdiversity, so there's the variation within populations that we can now sample without having to do culturing and isolation. And how you do that in terms of metagenomic data, I actually not really sure. I think we probably have to do some type of binning first and then assess the bins, but I think that good population geneticists or population ecologists might be able to design methods that are completely independent of that, that would start to say, we're just going to make a model of how a genome evolves in a context of a community, and how does that affect some metagenomic specific result, and you might be able to find evidence for that within the data set without ever doing binning. My instinct is that binning is going to help. Obviously, lots of what people here are interested in, and this is what launched metagenomics, is basically how it can help you do assays of what are they doing, which could not have been done before. And I don't really focus a lot on this, but I know a lot of people and what they said here, that's what they do. I think there are methods that need to be developed that are metagenomic specific for doing that. So we heard some examples of things that maybe people have done in a different context with genome sequence analysis, but now you can do with metagenomics. So taking 10 environmental samples, comparing them to all the genomes, taking all the orphans, and then building those into families, and then looking at something specific about those orphans that are shared between particular environments, or you can now correlate those to metadata and see if any of them are related to depth or other parameters. So I think there are a lot of sort of new metagenomic specific methods that will come out with functional predictions. And we actually haven't heard a lot about this here at this meeting, but I think that linking the who is out there to what are they doing is actually very important. This was basically what Jed Furman, the basis of a lot of what he was talking about was this ammonium oxidase gene and or processes, are they in those archaea, and how do you do this? And I think, even though he suggested that you've got to forget about looking at the data, you've got to do follow-up experiments, I think that there are a lot of things we can do with the data that will help find those patterns that might, obviously you want to follow them up with experimental studies, but might actually begin to tease apart. There is a metagenomic depth profile sequence data set that Dave Carl and Ed DeLong have generated already in the oceans. You could have done an analysis like he did with ERISA and with PCR of the different depths and looking at 16S distribution versus functional genes, and then trying to find cases where you could link the functional genes to some phylogenetic marker by assemblies or by other binning methods. So I think that again, is another area of metagenomics. And I forget who's Tim Hunkler who left because he thought we were all boring. I mean, I think the comparative metagenomic stuff is probably the most interesting and the hardest. And so I think I have I'm just going to skip over these things for a minute, because I think that the really hard part about comparative genomics relates to a lot of those challenges that I pointed out at the beginning. So if you want to compare 10 samples from different depths in the ocean and try and make some inferences about the communities of organisms in those samples and what their biological differences might be, how do you actually normalize, for example, between those different samples if the different number of sequencing reads is the simplest thing, or different sequencing methods, as I raised before, if they were generated by different people, or if the organism, if all of a sudden in one community nothing changes except one organism comes in that has a really large genome and doesn't contribute much of anything to the interesting biology or physiology in that environment, and maybe it replaces something else with that had a really small genome, all of a sudden the total proportion of your sequence reads relative to that community for functional genes that you might be interested in go down, but it might have nothing to do other than that an organism with a really large genome came into that environment. And so how do you actually do normalization across all of these variables? Yeah?
So I think eventually we're going to see more comparative method transcriptomics, which might circumvent what you talk about. But we're not going to see the genes that are, we're not interested in, like translocated and other kind of selfish things. But we're going to see what the bacteria are really using to cope with their environment.
I completely agree that that's going to be very useful. I think that there's absolutely untrue that we're going to see much of it relative to the amount of sequence data that we're going to see. So I think that, yes, that will help in many cases, but in most cases what we are going to see predominantly will just be sequence data.
But still, like sequence data from cDNA will help you.
Yes, so that, yes. You need to convince, so that's another thing. I mean, we need to convince the community, more people, to do things like that, because that will definitely help with, I mean, just any type of interpretation, but also just dealing with some of these complications of metagenomic data sets, because there certainly isn't a large, there aren't a large number of people doing that.
It's emerging of the law doing that now.
Well, but you know, the number of metagenomic sequence data, so DNA sequence data sets that are going to be coming out are, you know, tens of thousands compared to 10 or 20. I mean, so I think that the balance is probably a little off, right, in terms of what you can make the most interpretations of the data. But even when you have that, right, so even when you have that, you still have to try and figure out, even with the RNA sequence data, how to normalize across samples. And even if you did array, right, even if you actually had some targeted or studies, it's still very challenging to figure out how to normalize those with complex communities. It's even challenging with, right, the isolated genome organisms. But with complex communities, it's not entirely clear how to do that normalization, because the same thing can happen. One organism increases expression of all of its ribosomal proteins, and that's what you get, right? So you still have this problem of connecting the data sets that we get to the functional or distribution questions that we're interested in, that I'm not sure how to actually resolve that, other than testing the methods against simulations, testing the methods against ecosystems that we know something about, where you can say, I did this assay, but I know what the answer should be, and then see if the method actually normalizes correctly. So I think you can take, you know, a photosynthetic community, right, and do some study of it and say, we know that we've measured photosynthesis in the community. And now we take a really small sample of it, you know, do 1,000 RNA sequences and 10,000 DNA sequences. Can we actually detect, would our method have said this looks like a photosynthetic community? We're almost out of time here. We're supposed to end at 2:40, is that the...
I think get back down there absolutely no later than 2, so leave here at 10 up. So
Sorry.
we still have 10 minutes or so.
Absolutely no later than 3. Okay. So for the comparative stuff, there's a lot of different aspects to this. There's which we've seen in pretty much every talk today. There's the just clustering samples. Which samples are most similar to each other? Which has some of that's just helping you parse the data, and some of it is some real scientific or medical question, like about the Crohn's disease samples. Which are they more similar to? And so how you do that clustering actually is really important for making those inferences about the results. Correlations with the metadata. So if you want to do 10 environments versus pH and measure something about those the metagenomic data in those 10 environments versus the pH, and do a statistical regression of that result, if they all have different number of sequence reads and different phylogenetic distributions. So in Per Bork, in one of his papers, has this approach to trying to at least normalize, I wouldn't say normalize, weight by the relative abundance of different phylogenetic types in the environments to see if that affects the conclusions that you make. So in environment 1, you might have a big peak of some phyla that just happens to have, compared to other phyla, a huge increase in the number of ABC transporters, even though it's transporting the same amount of stuff as an organism with fewer ABC transporters. So if you analyzed ecosystems dominated by that organism, you would overestimate the amount of transport activity. And so if you can try and normalize this with sequenced genomes and say, well, this phylogenetic lineage has an overrepresentation of some gene, even though we might not think that it's related to the biology. Or ribosomal protein. So you can try and do these normalizations, but we don't really have any idea how to do that, because maybe in some cases those extra genes are functionally important, in other cases they're not. So I think that there are approaches, I think people have developed approaches to doing that, but I don't think we know how to actually weight these different results. And the same would be true for organisms with different genome sizes. So we might be able to figure that out by just looking at housekeeping genes that are single copy genes in genomes and use that to estimate the relative abundance of different types in the sample. But even when you do that, you're not weighting by the number of copies of the genome per cell, which is also enormously variable between organisms. So I think there are a lot of issues related to making inferences from comparative metagenomics that are going to require a lot of computational work to figure out how to actually do them and how to make sense out of them. Oh, and the last thing is, I do think, and we've seen lots of some examples of this in literature, that the comparative studies will, just like with comparative genome sequencing, be really useful for your analysis of a single community. So for helping annotate, obviously, the genes in a community, doing the comparative analysis will be very helpful for that. If you find your community has a bunch of genes that are normally unique to hydrothermal vent systems, it might tell you something, improve your functional annotation of the genes in that community. I think I'm just going to jump ahead. Well, that is the need. I think this gets to I think that if we think about what are some of the needs, obviously there are others, there's definitely a need for more testing of the methods that are being used, both through simulations, through model data sets. We could have, like they have with annotation of genomes, competitions, not that those have always worked out so well, but it would be interesting to try and generate model data sets that are metagenomic and then see how people can bin them, for example, or annotate them, or do things and have competitions that enhance the evaluation of methods. This was raised in the metagenomics report, and it's sort of obvious, it's come up in multiple talks here, but clearly metagenomic data analysis is multidisciplinary. There's need for people from ecology, from evolutionary biology, from statistics, from standard comparative genomics and computational biology, probably people from engineering to deal with the sampling aspects of it. I mean, there's obviously lots of different fields, math,
Don't forget computer science.
What is that?
Don't forget computer science.
Half of my lab, but and what?
And software engineering.
And software engineering. I mean,
5 to 10 bucks per 1,000 lines of code is what I usually hear.
Yeah. So no, but I mean, I think that is there's it is a little different than was the case for sequencing isolate genomes, because you've now added in oceanography and ecology and population biology and some aspects of sampling that were not present in previous fields. But again, what I was trying to emphasize were the things that were different than previous fields.
Documentation.
And what?
Documentation of everything.
That's the same is true, though. I mean, yes, absolutely, but that's nothing unique to metagenomics. It might be more important in metagenomics, I don't know, but probably not important in any software. I think that there is, I mean, I'm sure that there are pushes for this, but, well, standardization we've already talked a lot about. But for training and education, there's, I think we definitely need to emphasize this more. As far as I know, we've been looking around, there aren't, like, large number of metagenomics courses out there. There probably are some coming. I assume that Mitch isn't up here, but maybe MBL will have a metagenomics course or something like that. There obviously are integrated, so the microbial diversity course talks about this, and there are other courses that talk about it, but maybe there's a need for a community a course on metagenomics. And I put this last, but it's not the least important. The funding in the field for computational studies, we've seen in sequencing isolate genomes that the funding for computational work has generally been neglected quite a bit within that field. It is even worse in metagenomic analysis because the computational challenges are harder, and yet many of the funding agencies think you can just run the same program that they paid to develop for some isolate genome and you're done. And so I think that as a community we need to get together to provide more arguments for why funding has to be put into this if we want metagenomics to take off. So for example, the NIH has selected the human microbiome as one of the new roadmap initiatives. A component of that work will be metagenomic analysis. Most of that work will be what you would call environmental microbiology, but not all of it will be metagenomics. There are going to be unquestionably computational needs associated with that. We need to articulate before they dole out all the money and decide on what proposals to call for, the need for having computational specific programs within that to support the Human Microbiome Initiative. And the same is true for all the DOE biofuel initiatives, for and obviously the EU and the Japanese groups and all of the, I mean, I think that it's been a rule at every national and international level that the computational requirements are not as appreciated. And I think I really think that this is much worse for metagenomics. I think that there really is an expectation that a lot of this stuff is relatively simple to do, and we can just use the methods that were designed for all these other sequence genomes and push a button and we're done. So I think we should probably...
probably to wrap up, because you're going to have to get a summary down there.
I'm suddenly ill.
Everybody either sign up for a quick checkpoint like
So everybody needs to start a metagenomics blog and send the links to your congressional representatives.
No, then we need to build the super blogs that blogs all the other
Yeah, we can do that easily. We just need the blog out there to link to. That this blog is trivial.